So we talk about BLAST in this section. BLAST is most frequently used by informatics software. Almost everybody uses that. So BLAST stands for Basic Local Alignment Search Tool, and it is based on the same assumptions as FASTA, that those good alignments, they contain short segments of exact matches. So what we do over here is we try to locate ungapped similarity regions between the sequences. So we take the sequence for which we are doing BLAST, we call that sequence as query, and then we get some short segments within that query, we call them as words, and then we try to find the exact matches of these words within the search space, or we call it as search, or we call it as the database. So for a query, at first we try to find the high scoring words of length L. We start with a query sequence of length L, and then how many words we can have? It can be like L minus W plus 1, and for that we try to get the ones which maximum what we can get from them, so we retain them, and then we find the list of words scoring greater than some threshold. So longer is the good, so if words are longer and they are scoring better, then it's good, but that will kind of increase our search speed. So we can use different matrices, we score them, and we pick the ones that are scoring from some greater than some threshold. So instead of comparing each word of query with each word of the database, what we can do here is we can put the words which are similar, we can put them together in the groups. So we compare the words to the database, and then we find their exact matches, and then each match is then extended. So we get those matches, we retain the words scoring some greater than some threshold, and then for these matches we start extending these alignments in both directions. Where we stop, if our extensions keep on moving such a way that we are getting the scores greater than some threshold, then we are fine, but if they are scoring less than that, we stop over there. So here is how it works. We can see it again. So for each word match, the words are matching over here, and these are exact matches, and then after that we start extending these alignments in both directions for both of them, and we then retain those what we now name them as HSPs, high segment scoring pairs. So these pair, for example, is retained. This pair we can also retain as long as they are above a threshold. So in this way, now we took a query segment or a query sequence, and we get these high scoring segments within that, and then their pairs with all the segments within those database, and then BLAST returns the HSPs that have a score above a threshold, or the e-values less than some given threshold e-value. We'll talk about e-value and scoring scheme in a later on section. So here is the BLAST page. So basically it is there on NCBI web page, which is the largest repository as far as the biological data are concerned. So you can find some descriptions over there. You can go to NCBI website, and there is a link NCBI BLAST. You can click it, and then it will lead you to this page. So here you can study different aspects of this software and different facilities you can enjoy over here. We can also have assembled genome. Down there is a list of genomes and their gene models, which you can pick them, make them a database, and compare your sequence against that. So all of them, they are underlined down here. Their links are provided. So if you want to run BLAST, you can scroll down, and then there is a button. If you click that, it will lead you to this page. So here you go, and here is the page where you can put your FASTA sequence over here in this window. You can choose those sequences over the website, or you can browse and upload your own file here from your local machine. You can align 2 or more sequences, so in this way you can also do a pairwise comparison over here. So you put your sequence over here, you select your parameters, and down below there is a go button. So when you click it, it will then start doing BLAST for your sequence. In this page, you see at the top there are other options also. If you want to do nucleotide BLAST, here you go. for proteins, and then there are other options. We'll talk about them later. So here comes your results in a graphical display mode, where you can see your sequences at the top, and then those most closely related sequences are lined up, and then these colors, they mean something, just like FASTA. So after the blast is performed, you get the results in different ways. So this is one of the way, which is the graphical output. So here you see those sequences are represented by these lines, and these red lines means the sequences are highly related to your sequence. So their scores are given over here. If the scores are above 200, they are given red color, and so on. They are different colors, 80 to 200. So less than 40, they are colored black. So they will be down there. So they are arranged in an order also. So highly correlated sequences are at the top, and then so on, it decreases downwards. You can get the output as hit list also. So it tells you at the top of this thing. So basically it provides you the sequence itself. So first, if you give any sequence, it will go to the NCBI and try to find out what you have given to him. You just gave the sequence, so it get that sequence, and on the top of the list it showed us where obviously this similarity is 100%. So this one was your sequence, so it is identified as Arabidopsis thaliana, F-box, blah blah blah, whatever this is. Its score of this alignment is 641, and then the total score, which is obviously 100%. So obviously out of 641, it's 641. So here is a column that says query coverage. So in this, it tells you like how much portion of your query was covered in the alignment. So were every section or every segment of your query was searched? Yes, so it is 100%. So for example, your query is this much. Sometimes we see that in the alignments, these parts are only present while other parts may be missing. This may be present. So this, since it's a local alignment, so it will do the alignments within some local regions of similarity. So query coverage will tell us like how much of your query has been covered in the alignments. It comes up with a e-value, that is the correlated with the probability of finding this match, right? What is the true probability of this match as compared to the random probability or by chance probability. So in that case, this e-value is really important. Lower is the good. So this is small e-values are preferred. Identity is 100%. That tells you that this sequence is 100% identical. There is another phenomena I would like to just explain over here, that is called similarity. So identity is exact identity. Sometimes amino acids are similar, so in that case we can say we have similarity. Here is one of the segments. So we have query at the top. So nucleotide number 1 of your query matches to 6677 in your database, whatever the sequence was. So it's 677 position was matched to first position of your query, and these vertical lines, they tell us about the exact matches. If we have some gaps, we will see them. If we have mismatches, we will have some symbols like stars, etc. over there. There are no gaps as we see over here. So BLAST improved overall search speed and put database searching on a firm statistical foundations. And this is an important tool, which is a local alignment search tool.